Welcome to another episode of the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. My name is Darleen Sandoval, and as always, I'm joined by my podcast co-host, Dr. Kirk Habiger and Dr. Kevin Williams. How are you all doing today?
Doing good, Dar. You?
Hanging in
Doing
there?
good.
All right.
Weather's
Kevin?
been decent.
Doing good. Yeah. Yeah. It's been a cool summer so far. Probably going to get some heat pretty soon. But what about you in Alabama and Colorado? What's going on?
We've had rain for weeks,
Oh, wow.
like
Yeah.
every day, and cooler than normal. So yeah,
Oh,
the grass
that's
is green still.
Yeah, we've had a very weird spring. First it was hot and dry, and now we're getting a lot of it's almost like it's monsoon season. We get the afternoon clouds and showers and stuff. So had hail a couple days ago.
Oh,
Yeah. We're recording this the first week of June. Do you all have summer plans besides continuing to push the boundaries of science?
I do. But first, I want to hear where Kevin has been.
Oh, yeah.
Because he was unavailable for every single date that we proposed.
That's right.
That's right. Yeah.
Where were you, Kevin? What were you up to?
It's my first day back on this time zone. I was 12 hours shifted, so I'm adjusting so far fairly well this day, but we'll see how that goes. I don't know if actually my words are coming out of my mouth appropriately or not, but yeah, I was over on the other side of the world. I was in first Cambodia and then in Korea.
Oh, wow.
And that was pretty cool. I'd been to Cambodia 24 years ago, and so it was neat to see the shifts and development of the country and the growth of the different cities. Went to Phnom Penh and Angkor Wat. So Siem Reap is the city there, and seeing those temples. It's just going back, I don't know, multiple centuries. I think it was back in, I think they were saying like 600 or something. Those temples were built, maybe into I don't know. I could be speaking completely incorrectly there, but it's the temples are so impressive over there. And then in Korea, I had a little bit of work to do with going to a conference and experiencing a bit of the cities and culture there, more so than I've done in the past, because a lot of times when you go on these conference trips, I know Kirk and Darleen, you're aware of them, that you end up going to the conference and then you rotate around from city to city talking to universities. And so a lot of times you don't have a lot of time to experience the city. And so I spent a little bit of extra time with some of the cultures and the different regions. So that was cool.
Well, that's very cool. I cannot top that.
Yeah, me neither.
We
might go to Maine in August.
Well, coming up this weekend, we'll be in New Orleans for the American Diabetes Association's meeting. So that'll be pretty fun.
That'll be good.
It won't be quite, I guess, Cambodia or Korea,
No.
but New Orleans has great culture,
has
great food.
Yeah. It's a great place.
Yeah.
All right. So I guess we should get to work. I guess we have a little bit of a disclaimer to make for today's episode. I swear to all of our listeners, we do try to be unbiased in our selection of authors that we interview, but it just happens today that all the authors we're interviewing are close friends and colleagues.
Sure, but I think, my mom always told me it never hurts to be nice, right? So,
all right. So Kirk, tell us what we have on board.
That's right. This month, we have another great set of articles. We're going to start with June's paper of the month, which was Interrupting T-Cell Memory, Ameliorates Exaggerated Metabolic Response to Weight Cycling. We were joined by corresponding author, Dr. Alyssa Hasty, who is now professor at UT Southwestern in Dallas, and first author, Dr. Jamie Garcia, who conducted this work as a postdoctoral fellow while in Dr. Hasty's lab when she was at Vanderbilt. I know I personally have been interested in weight cycling since Cincinnati, right? When I was a postdoc, and seeing this immune-based mechanism layered onto that known physiology was really cool.
Yeah, it's a really neat model.
Following that, we discussed gut-derived FGF15 modulates lean mass, bone, and bile acid responses to weight loss with corresponding author, Dr. Randy Seeley, professor of surgery, and first author, Nadejda Kramer, who's an assistant professor, also in the Department of Surgery at the University of Michigan. So always good to talk to friends.
All right. And Kevin, what do we have going on for Sweet Talk?
Yeah, today we're talking to the 2010 or 2010 ADA Outstanding Scientific Achievement Award winner, Dr. Martin Myers. Dr. Myers directs the Caswell Diabetes Institute at the University of Michigan, and he is a former editor-in-chief of this very journal, Diabetes. His lab has performed foundational work dissecting how leptin receptor signaling promotes weight loss, food intake regulation, regulation of puberty, and many other facets of leptin signaling. And more recently, he's been studying how circuits in the brain can explain how GLP-1 based drugs induced weight loss and why separating weight loss from nausea it might be harder than the field had hoped. We'll get into all of that, so stick around and find out more. And I should mention, like in the interview, you'll hear it, but I mentioned that when I was in graduate school, one of the very first papers that I had to highlight in graduate school was Martin's work, Sarah Bates' paper. And just found it fascinating, how your career trajectory or whatever arc happens that ultimately I'm interviewing or we are all interviewing and talking to this really foundational investigator in the field. And I don't know if he appreciated that kind of comment because it does date people. So I don't know if he felt old when I was telling him in grad school I'm highlighting a paper that he did, but it was really a neat moment to be able to talk to Martin and have that reflection. So I think it's interesting what we all do on this podcast, talking to these people in science and
Yeah.
having that opportunity.
And you know what the coolest thing is that they're all really cool people too. So
I was going to
Yeah,
say,
absolutely.
great scientist and actually a decent human as well.
yeah. Yeah. Awesome. Well, let's get started. Today we will be discussing the June paper of the month titled Interrupting T-Cell Memory Ameliorates Exaggerated Metabolic Responses to Weight Cycling. We are joined by corresponding author Dr. Alyssa Hasty, who is now Vice Provost and Senior Associate Dean of Faculty Affairs at UT Southwestern in Dallas, and first author Dr. Jamie Garcia, who conducted this work as a postdoctoral fellow in Dr. Hasty's lab when Alyssa was at Vanderbilt. Welcome, Alyssa and Jamie, and thank you so much for sharing your work with us today.
Well, thank you so much for having us. And we're just delighted that our paper was selected as the paper of the month and excited to share some of the data and information and conclusions from the paper with you.
Awesome. So we like to start with an icebreaker question. And I have a new one for you. We have like sort of a list of icebreaker questions that we go through, and I have a new one for you today. So what is a scientific fact that still blows your mind? And doesn't have to be related to adipose tissue or the immune system or anything like that. It can be anything.
So I don't know why this really caught my attention. It was something published a few years ago, and it was about how the lymphatic system in the brain clears out waste while you're sleeping. And I just thought that was so fascinating. I think the lymphatic system is not given enough credit for all of the things it does for us. And I just thought that was really cool.
That is cool. Jamie, do you have one?
Yeah, mine straddles, I think, both science and a little bit of my passion for endurance sports. But I was really surprised to find that cross country skiers possess some of the highest VO2 maxes ever recorded. Growing up in California, that was not a sport that I was super familiar with. So that was pleasantly surprising to find.
Yeah, those guys have some big engines, man. It's pretty
Darleen, you
cool.
and Jamie, you two would have a lot in common in thinking about endurance sports and long distance running.
As
We
as
should
would
connect
Kirk. Kirk
sometime.
is a runner as well.
Oh,
Yeah.
okay.
Amazing.
Yeah,
Excellent.
It's a contagious affliction.
it is.
It's only a matter of time.
Yeah.
All right, Alyssa, let's turn now to the work. And I'm wondering if there are any other authors or collaborators that you'd like to acknowledge as you completed this article.
So I will actually mention them as I go and as I describe some of the work. But in particular, I'd like to mention Dr. Nathan Wynn, Dr. Matthew Cottam and Dr. Heather Caslin, who were either students or postdocs in my lab at the time that they worked on this project with Jamie and a few others in the lab.
So now maybe you can give us sort of an overview of your research program and how this specific project fits into it.
Absolutely. My research program was broadly focused on how obesity and, importantly, weight history shape immune function and metabolic disease risk. We approach this question from an immunometabolism perspective with a particular interest in how immune cells within adipose tissue are remodeled by overnutrition, weight loss, and weight regain, which we refer to as weight cycling. We are also interested in whether those changes are reversible. Chronologically, this work began over a decade ago with a relatively simple but underexplored question: what are the metabolic consequences of weight cycling? In a 2013 study led by a student, Emily Anderson, in lab, which was also published in Diabetes, we showed that repeated cycles of weight loss and regain worsen glucose homeostasis compared to sustained obesity and are associated with increased accumulation of T cells in adipose tissue. That paper was one of the first to suggest that adaptive immune cells might be responding not just to adiposity, but to prior metabolic exposures. From there, as technologies evolved, we pushed the work in more mechanistic directions. Using flow cytometry and eventually single cell approaches, we began asking whether obesity-associated immune phenotypes truly resolve with weight loss. A major step forward came in 2022 when Matt Cottam, now a faculty member at Vanderbilt Medical Center, and Heather Caslin, now a faculty member at the University of Houston, partnered on a study showing that adipose immune cells, particularly T cells, retain transcriptional and functional features of inflammation even after significant weight reduction, and that these features are amplified with weight regain. That work introduced the idea that immune cells carry a form of metabolic memory of prior obesity. This immune memory concept became a central theme of the lab, and we next asked whether it extended beyond adaptive immunity. In a 2023 study, Dr. Caslin led work demonstrating that adipose tissue macrophages develop features of innate immune memory during weight cycling, including metabolic and epigenetic reprogramming that primes them for exaggerated inflammatory responses upon re-exposure to excess nutrients. Methodologically, this work integrated single-cell RNA sequencing with chromatin accessibility and metabolic profiling, and it provided a mechanistic framework for how prior obesity can have durable immunologic effects independent of current body weight. In parallel, we've been careful not to view adipose tissue in isolation. Matt Cottam and Nathan Wynn, who is now a faculty member at Vanderbilt Medical Center also, examined how weight cycling impacts systemic metabolic regulation, including pancreatic beta cell function and insulin secretory capacity, highlighting that immune memory in adipose tissue likely propagates signals to other organs involved in glucose homeostasis. And that work also was published in Diabetes in 2022. In the current study, Dr. Jamie Garcia wanted to test whether interrupting adaptive immune cell memory formation could interrupt the metabolic detriments associated with weight cycling.
Awesome. That was a great backdrop for setting up the paper. Jamie, do you want to kind of step back and tell us a little bit more about maybe some of the nitty-gritty details on how you generated the models, so obesity and the weight cycling model, and then we can get into the first couple of figures.
Sure. As Alyssa mentioned, our lab had a really well established diet paradigm that many of us have used in our papers throughout the past decade. And so we generated lean, obese, and weight cycled mice using that same diet paradigm where we place mice on a 27-week diet paradigm that changes every 9 weeks. So, for example, lean controls were maintained on low-fat diet for the entire 27-week period. The high-fat animals were started on low-fat diet for the first 9 weeks, and then for the second 2 9-week periods were placed on high-fat diet. And then our weight cycling group started on the high-fat diet, was transitioned to low-fat, and then for that final 9-week period was placed again back on that high fat diet to regain weight. This design is really powerful in that it allows direct comparison of both immune and metabolic outcomes in mice with similar backgrounds, but distinct controlled histories of dietary exposure and also weight change.
Just out of curiosity, how old were the mice when they started the studies?
Yeah, the mice were anywhere from 8 to 9 weeks of age.
Awesome. All right. So let's talk about your single cell RNA-seq, at least the first batch. I know you did kind of a couple of cohorts or a couple of runs on this. So you were focused on a specific type of immune cell within the adipose tissue. And so for folks that don't know much about the immune system, for scientists that don't know much about the immune system, can you describe those? Kirk just raised his hand for those who can't see the video.
It's definitely
Um,
me.
Can you describe those immune cells and why they were a particular important population to study?
Absolutely. And similar to Kirk, I was also not trained as an immunologist. So, in coming to Alyssa's lab, this was a new area of biology for me to explore as well. And in the paper, the core biological question we were trying to address is whether adipose tissue remembers prior obesity or weight change in a way that worsens metabolism during weight regain. Long lived immune cells, especially T cells, are prime candidates to address this question because they can persist after an initial stimulus and respond more strongly upon re-exposure. So in profiling adipose tissue immune cells, and really specifically these T cells, allows us to quantify which immune populations change over these various diet conditions, such as obesity, weight loss, and weight cycling, and also allows us to test whether T cells show signs of expansion and memory-like persistence. So are these T cells the key to this obesogenic memory that we seem to be observing?
And that's exactly what you kind of conclude from the data, right? So what were the data that really led you to that conclusion?
Yeah, what really convinced us that T cell memory goes up with weight cycling was a couple of things pointing us all in the same direction. When we did the single cell profiling of the CD45 positive immune cells within the adipose tissue, we saw that the weight cycled mice had a noticeable shift towards a CD8 T cell population that's a marker of that memory phenotype, especially compared to the lean counterparts. As a brief reminder, T cells are a type of immune cell that help the body detect and respond to threats by recognizing specific targets and coordinating immune attacks. Some T cells can become long lived memory cells, meaning they stick around in the body after an immune challenge and can react faster when that same target or antigen is represented. Then, when we looked at TCR clonality, we saw even more evidence of clonal expansion. As a primer, TCR clonality is like seeing the same barcode on lots of T cells, which usually means one T cell found a target it could recognize and then made lots of identical copies of itself. What stood out is that this clonality wasn't just an obesity thing, but it persisted and was even higher in the weight loss and weight cycling context. So, in putting these observations together, the increase in memory like CD8 states and the expanded clones suggested that adipose tissue is carrying this immune memory through weight cycling. And that's what pushed us to studying the CD27, CD70 axis, which I think we'll get into in a little bit.
So I want to follow up on the clonality question here. For someone who's not an expert in immunology, first of all, how do you measure this? And then give us a little bit more about what that means.
Yeah, so in order to look at the clonality or these barcodes that were being populated or presented on the T cells, we kind of paired what's called VDJ sequencing with our single cell analysis. And so VDJ sequencing is a method that reads these unique genetic barcodes each T cell uses to make its receptor. And because every T cell has a different barcode, this lets us track which T cell groups have expanded and whether the same clones persist over time or within certain diet groups. Yeah.
So you had some surprising results in the weight loss group itself, right? Can you explain those for us?
That's correct. So we know from previous work that even though weight loss often brings metabolic measures back towards normal or baseline, the adipose tissue immune compartment doesn't fully reset. And that was really surprising to us. So, in other words, the mice can look metabolically improved after weight loss, but we still see traces or markers of prior obesity in the actual immune signature. Specifically, the adipose immune profiling suggested persistence of a more obesity experienced obesity experienced T cell state with persistent clonal expansion in CD8 T cells from our weight loss group, rather than a complete return to lean like baseline T cell signature. This immune lag is exactly what made us think about, again, this memory mechanism that could matter when weight is regained.
All right. So you mentioned the CD27, CD70 axis. So tell us more about it and how you tested its role in adipose tissue function with weight cycling.
The CD27, CD70 axis is basically one of the key go signals or go pathways that helps T cells fully turn on and activate. And it's especially important for building and maintaining T cell memory. Because the CD27, CD70 axis is involved in immune memory formation in other contexts outside of obesity and diet, we thought we could use it more as a tool or resource to interrogate the role of immune memory in weight cycling. We were fortunate enough to have collaborators at Vanderbilt that had CD70 knockout mice. So they're whole body CD70 knockouts that we used as a tool in this study, again, to interrogate immune memory.
Awesome. So tell us about the metabolic phenotype of that mouse, especially when they were placed on this weight cycling paradigm.
To test whether that a CD70, CD27 pathway mattered, we used the knockout mice and ran them through the exact same diet paradigm that we did wild type controls, as we've done many times over throughout other papers and projects in the lab. While there were no differences simply by being placed on lean or low fat diet or high fat diet only, what we saw that was with loss of CD70 and that second bout of weight gain, the mice were actually protected from that second hit glucose intolerance that we know characteristically shows up after repeated bouts of weight cycling. So in the past, in wild type mice, when they're placed on our weight cycling diet paradigm, after that second exposure to high fat diet, they have worsened glucose tolerance. However, in the CD70 knockout mice, they did not show that same phenotype, again, suggesting that they were protected.
Great. So lastly, you examined the immune cells within the CD70 knockout mice that had undergone weight loss. Can you tell us what you found there?
Yeah. In the CD70 knockout mice that had lost weight, the big takeaway was that their adipose tissue didn't keep the same immune memory of obesity that we're used to seeing in wild type adipose tissue. When we looked at immune cells in the fat, there were fewer of the T cell clusters you'd expect if a long lived, organized immune shift or immune response had formed. When we zoomed in and really focused on the T cells specifically again, there were fewer of those CD8 T cells, the one that most strongly carry that persistent memory-like signal as compared to the wild type animals. And overall, there was less evidence that a small number of T cell families had expanded and stuck around long term. So in summary, taking away CD70 blunted that long-lasting T cell imprint in adipose tissue after weight loss.
Very cool. And I guess as a follow-up to either of you, we're living in an age of treatable obesity. Do you think any of the current or maybe even upcoming peptide-based therapies might modulate this immune component?
It's definitely something that we've been interested in. We haven't tested that experimentally, but it's a critically important question because as humans start to lose dramatic amounts of weight on these new drugs, and we know that oftentimes they regain that weight if they stop taking the drugs. The human component of this really extreme bouts of weight gain, weight loss, and weight regain could be impacting the immune system in the same way that it's doing in these mouse models. And so we do think that it's important to learn more about how this is impacting the immune system and the relevance toward humans with an eye toward thinking more holistically about weight loss and trying to make sure that we're understanding more than just what's happening metabolically, but also what's happening immunologically.
Very cool.
All right, Alyssa, are there any other key takeaways from this work that you'd like to leave us with? And I know that you have moved on to a very important administrative role at UT Southwestern, but are there any key questions you'd like to see the field address moving forward?
I think I sort of addressed part of that. What I think are some future questions that are very important for the field is for us to understand the relevance of this in humans. But from a translational standpoint, this work really enforces a key message from sort of the entirety of the program that I was describing to you earlier, which is that weight history matters. So, in addition to thinking about the metabolic and immune consequences of weight cycling, oftentimes when we go to a doctor, they'll take our weight, but they don't ask us about our weight history. And perhaps that's a particularly important question for a physician to know as they think about the treatments that we might need, in particular for cardiometabolic diseases. So I think this idea that the immune system doesn't reset with weight loss and that repeated weight cycling induces this durable cell-specific immune reprogramming is particularly important for us to understand. And so from our work and what I hope will be continued by others, both my own trainees as well as other labs around the world, identifying new immune populations and shared programs across different cell types, we hope that this can inform strategies to mitigate the long-term immunometabolic consequences of obesity, especially in this context of weight loss interventions that are increasingly common.
Alyssa and Jamie, we want to thank you for joining us today and for sharing your expertise and most importantly, for discussing your recent work on the roles of immune cell memory in weight cycling. I encourage our listeners to read the full article in the June issue of Diabetes.
Today, we will be discussing a featured article from the June edition titled Gut-Derived FGF15 Modulates Lean Mass, Bone, and Bile Acid Responses to Weight Loss. We are joined by corresponding author Dr. Randy Seeley, who is the Henry King Ransom Endowed Professor of Surgery, Professor of Internal Medicine and Nutritional Sciences at the University of Michigan School of Medicine, where he also serves as director of the NIH-funded Michigan Nutrition Obesity Research Center. So shout out to another NORC.
Go
We're
Blue.
also we're also lucky to have first author Dr. Nadejda Kramer, who is assistant professor of surgery at the University of Michigan School of Medicine. Welcome, Nadejda and Randy, and thank you so much for joining us today.
We're so happy to be here. Right, this is super fun.
Thank you for having us.
Dar,
All right.
I think we should start with the disclosure, and that is that Randy has mentored all three of us over the course of his career.
I'm not
and
making
he's still
that
alive
claim.
somehow.
It's a subtlety that I think most of the audience won't appreciate, like how difficult I make it.
So with that, we usually start our podcast. I'm sure you've listened to many episodes, so you know this. We start our podcast with an icebreaker question. And so for the first time in podcast history, we're going to ask two different icebreaker questions. And so the first one for Randy, who, by the way, just won a mentor award from the American Gastroenterological Association. Of all the years of being a mentor, who is your favorite mentee?
Wow. I mean, isn't it obvious, right? That that it has to be Kirk. I mean,
There it is. My first win.
I think it'd been more fun if you asked me like who is my least favorite.
Oh, yeah, that's
also
true.
I was gonna say, is that question number 2?
All right. So, how about giving us a fun fact about yourself that has nothing to do with science? Nadia, we'll start with you.
That has nothing to do with science.
I guess we'll give you a little
I'm
leeway.
really good at cross stitch.
Oh,
I
okay.
make ridiculously good sourdough bread.
Oh, okay.
Oh.
Yeah.
Nice. Randy, can you confirm that?
I can confirm that there's much baking, right, that shows up in the laboratory that is quite delicious, right? So that helps a lot.
All right, Randy, do you have one for us?
Well, I always refer to Drucker, refers to me as having the best Seeley index in science, which is the ratio of your H index to your golf handicap.
That's pretty
I
good.
don't think
That's
it's an
really
official
good.
number, right? But
Yeah.
that's
I like that one.
It's a good
All
one.
right, we should probably get to the work. So, Randy, as we start, are there any other authors or collaborators that you'd like to highlight that are specific to this work?
Nadia, do you want to jump in? Who do you want to highlight?
I think a big contributor to this study was Orman McDougall and Zero Lee. They were really instrumental in a lot of the skeletal remodeling studies that we showed in this paper.
Very cool. All right, so Randy, maybe start with an overview of your research program and then how this specific project fits into it.
Yeah, so our lab's really interested in gut-brain communication and how that gut-brain communication be leveraged for therapeutic purposes, particularly for weight loss. So we spent a lot of time thinking about bariatric surgery, and we spend a lot of time thinking about gut hormones, and that's where this comes in, right? So we got interested in this gut hormone because Nadia had found, right, that FGF15 is greatly increased in expression after bariatric surgery. And so FGF15 is a weird gut hormone, right? Its timing is very different, right? It takes several hours before it rises. It has a clear role involved in bile acids, but the receptors are more broadly expressed than that. So we were really interested in understanding how FGF15 fits into the gamish, right, of signals that come from your gut, talking to your brain and other organs.
All right, awesome. Nadia, let's get a little bit more into the article.
Mhm.
So you investigated the regulatory functions of intestinal-derived FGF15 on several different metabolic parameters, and as the title implies, you looked at lean and bone mass, glucose tolerances, and changes in bile acid and lipid parameters. Why don't you tell us a little bit more about the mouse model that you used, and also maybe the dietary and pharmacological stimulated weight loss methods that you used as well?
Yes, we used an inducible mouse model where we ablated specifically intestinal FGF15 in adult mice. We used two different weight loss interventions to induce weight loss. All of the mice were subjected to high-fat diet feeding for 22 weeks to induce obesity. And after this, they were randomized into switching the mice from high-fat diet to chow diet to induce weight loss, or maintain the high-fat diet feeding, but treat the mice with semaglutide to induce weight loss.
And what was the rationale for this particular experiment and maybe these models? You had previous experience with the intestinal knockout, right?
Yes, exactly. So really the rationale of the study came directly from our previous work, looking at the role of the gut hormone FGF15-19. So FGF15 is the mouse ortholog, FGF19 is the human ortholog, and just overall its role in weight loss physiology. And as Randy mentioned in earlier studies, we were really interested in why some forms of rapid weight loss intervention really cause substantial reductions in lean mass, where other interventions cause not such a substantial reduction in lean mass. And also, as Randy mentioned, we knew from our studies and from other clinical studies that bariatric surgery does induce increase in circulating FGF15 and 19 levels. And in our previous publication, we looked at vertical sleeve gastrectomy in a mouse model where we used the same FGF15 knockout mouse model. We ablated FGF15 specifically from the intestine. And we found that intestinal FGF15 does protect against lean mass loss after surgical weight loss. And this was really an important observation because it suggested that FGF15 was doing more than just regulating bile acid synthesis. It might also participate in this broader gut to liver to lean mass communication. On the other side, looking at obesity, we have also reported that in obesity, and also when you add comorbidities to obesity, FGF19 levels go down. Now, when human subjects with obesity have rapid weight loss from a very low energy diet, those FGF19 levels overall recover. So we see this increase in FGF19 levels after weight loss. And again, in another recently published study, we saw that particularly in human patients with obesity and type 2 diabetes, the response, that increase in FGF19 levels, does predict the percent lean mass loss these humans will lose when subjected to very low energy diet-induced weight loss. So all of this put together was really the rationale behind this current study, and really raised several new questions. So first, we wanted to ask the question whether the role of FGF15 on lean mass regulation and skeletal regulation are really dependent on a particular weight loss intervention. And this is how we design the two different weight loss interventions, switching, going from high-fat diet to a dietary induced intervention, switching the mice to chow diet, or maintaining them on the same high-fat diet, but inducing weight loss with semaglutide.
Also interesting, just given there's a lot of opinions about semaglutide-induced lean mass loss, right? So
Yes.
so I think also interesting along those lines.
Yeah, my TikTok feed is full of opinions about semaglutide with a range of expertises that are associated with that.
Yeah.
And this is definitely a point of contention, right? Which is that these drugs are associated with lean mass loss, not muscle loss, lean mass loss.
Yes.
And there are definitely people out there who say nobody should take them because of the dangers that might be result from that lean mass loss. And that's certainly part of why we did the study the way we did, to try to understand what the dynamics of that lean mass loss is, what are the things that might contribute to it, and what you might do to mitigate it if necessary.
So for the record, Randy saying my TikTok feed is full of was not on my bingo card for today.
Well, again, obviously, you're just not keeping up with the kids,
Oh,
right?
I'm not.
I am hip and down with the kids, Kirk, right? Right?
You always have been, Randy. 100%. Nadia, I would love for you to follow up on the lean mass thought and maybe detail the specific findings of this paper and the difference between dietary weight loss and semaglutide-induced weight loss.
That was one of the most interesting distinctions in the study. What we found is that intestinal FGF15 appears to be protective for lean mass, specifically in the context of diet-induced transition, again, from high-fat diet to standard chow diet. In that setting, wild-type mice preserved lean mass better, where FGF15 knockout mice lost more lean mass. Now, when weight loss was induced pharmacologically with semaglutide, while the mice, again, remain on high-fat diet, that FGF15 dependent protection essentially disappeared because both wild-type mice and the intestinal FGF15 knockout mice lost comparable amounts of lean mass. My interpretation is that semaglutide engages a different physiological program than diet switching. We think that this underlies the difference between the preservation of lean mass between both rapid weight loss interventions.
Right. And you also observed changes to the skeletal remodeling as well, right?
Yes, we did. The skeletal findings also reinforce that these interventions are not biologically equivalent. In wild-type mice, semaglutide-induced weight loss was associated with changes in trabecular bone. Particularly, semaglutide reduced trabecular bone volume fraction in the trabecular bone mineral density in wild-type mice. The trabecular thickness was also reduced in both wild-type mice and the FGF15 knockout mice that were treated with semaglutide, and that is in comparison with chow-fed wild-type mice. We did not see any changes in cortical bone parameters, but we did see changes in the bone marrow adipose tissue, or the BMAT. Semaglutide-treated wild-type mice both had an increase in BMAT in the proximal tibial, as well as the constitutive, the regulated BMAT in the proximal tibial, as well as the constitutive BMAT in the distal tibia. They were both increased, but that increase was not observed in the FGF15 knockout mice. So overall, the skeletal response really seemed to depend on different interacting factors. We have the weight loss method that influenced the skeletal remodeling. We believe the underlying diet also played a big role. And of course, the presence or absence of intestinal FGF15 also contributed to that skeletal remodeling after rapid weight loss.
Right. And then you did a cool thing where you released these animals to untreated free feeding, right?
Yes.
And do you want to tell us kind of about those findings as well?
Yes. So this study really came, the idea of doing the discontinuation and looking how the body composition responds when we discontinue semaglutide treatment really came after we had completed most of these studies. It was, as Randy mentioned, it was a very important topic. It has been a very important topic. And because semaglutide was so, had such a strong response in reducing body weight and reducing lean mass in both wild type and FGF15 knockout mice, we asked the question whether the recovery of body weight, of the body mass and the lean mass depended on FGF15. The recovery, again, after discontinuation of semaglutide. So just as you said, we repeated the experiment where we treated high-fat diet, wild type, and FGF15 mice they were on high-fat diet and semaglutide for 26 days. And after that, we discontinued semaglutide and we continued to follow their body weight and body composition. And again, another really key point, I think, from the study in this discontinuation experiment, we saw that all mice return towards baseline pretreatment levels in body weight, in fat mass, and in lean mass. So the semaglutide effect on the body composition was reversible. I think two important implications. First, the semaglutide did not produce a permanent loss of lean mass in our mouse model, in both the wild types and the FGF15 knockouts. The effect was really tied to the ongoing treatment. And I think this data really highlights the importance of what happens after pharmacological weight loss therapy is stopped, and really supports the idea that the continuation of therapy or introducing some other lifestyle interventions are really needed to maintain the body composition.
Yeah, I think this goes back to the TikTok expertise, right? Where lots of folks believe that you're going to regain the weight, but you're going to regain it all as fat mass, and you will not regain the lean mass. And that, again, in these mice, that just doesn't turn out to be true. And I think that's a risk that has been overstated. The one caveat to this, right, is the animals are relatively young, right? I think the issues about individuals who are older and might have already reduced lean mass, even with concurrent obesity, I think we need to know more about what actually happened. But what I would say also, right, is that there's not functional deficits that are created, right? So recent work has shown that there are not strength changes or losses that are associated with that lean mass. And again, everybody needs to understand, right? What gets measured and reported in these clinical studies is lean mass. It's not muscle, right? So it's a confluence of lots of other tissues, right? And that sometimes, I think, confounds the interpretation of it in ways that most of the TikTok experts don't seem to know.
Or it's even worse, right? It's a 2-component model. So you're fat-free mass. You're not even
exactly right. Like
right.
It's a DEXA scan, right? It's an X-ray, right? That you're using to measure this, right? And it clearly overstates the loss, right? Because at least if you think of it as muscle, because
Mhm.
the livers get smaller, and that gets
Yeah.
counted as part of fat-free mass,
Yeah.
even though that liver getting smaller is a good outcome rather than a
Yeah.
bad outcome.
Absolutely.
Nadia, let's switch gears a little bit, and of course FGF15 is really important in bile acid production and sort of circulation. So can you talk a little bit about what you found in terms of the link between bile acids and the changes in lean mass?
Yes. We did have a hypothesis, and I need to preface this by saying that we did not test this hypothesis in the study, but we have ongoing work that is investigating this mechanism a little bit more closely. But our hypothesis is that not having intestinal FGF15, as well as semaglutide-induced changes in bile acid composition, really lead to changes in the secondary bile acid pool, and in particular, the deoxycholic acid. And our hypothesis is that this increase specifically in the secondary bile acids, or maybe even more specifically to the deoxycholic acids, may contribute to the lean mass loss. So just a little bit briefly, what deoxycholic acid is it's secondary bile acids that is generated by microbial modification of primary bile acids in the intestine. And in our study, semaglutide increased circulating levels of deoxycholic acid. Importantly, this happened both in wild type mice as well as intestinal FGF15 knockout mice, which I believe mirrors the fact maybe that semaglutide reduced lean mass in both, regardless of FGF15 status. And again, our study did not directly investigate this link, but we did reference a couple of other studies that have looked at this mechanism. Studies have shown that having very high levels of bile acids, particularly these hydrophobic, more toxic associated bile acids, lead to skeletal muscle dysfunction by by inducing mitochondrial dysfunction in skeletal muscle. And another study has shown that treating sarcopenic mouse models with exogenous FGF19 can rescue those changes, that decrease in lean mass. What we discussed in the paper is that currently the hypothesis is that this could be an FGF15 specific, FGF15 to lean mass communication, or these changes that we see in lean mass could be due to changes in bile acid composition. And again, this present study did not directly investigate this, but again, we are following these with some exciting new studies to try to answer this specific question.
Excellent. Semaglutide may also act as a bile acid sequestrant. You want to talk about that a little bit?
Yes. So this idea really fit well with our observations, again, with important nuance. So prior clinical work has shown that GLP-1 receptor agonists behave somewhat like bile acid sequestrants. And in our study, semaglutide did increase total bile acid content in the cecum, which really supports this concept. However, in our study, semaglutide did not change the levels of the total circulating bile acids, but instead it altered the composition of the circulating bile acid pool. And I believe this distinction matters. Again, the sequestration-like effect could retain more bile acids in the intestinal lumen or cecum in changing that exposure to the gut microbes. And that really could promote the conversion of some of the primary bile acids into secondary bile acids, including the increase in deoxycholic acid, such as what we report and what has been reported in clinical studies with GLP-1R agonists.
So, Nadia, let's talk a little bit more about the bile acids. And you didn't just look at deoxycholic acid, but you actually profiled the pool in general. What can you tell us about the bile acid shifts that occur in response to semaglutide, and maybe which of those are FGF15 dependent?
That's a great question. We saw very distinct changes in the bile acid pool. So after weight loss interventions, the pattern of bile acid composition really depended strongly on whether the mice were switched to chow or treated with semaglutide. The chow-induced weight loss tended to normalize some bile acid species, but in contrast, semaglutide maintained or amplified some very distinctive bile acid changes, particularly in the FGF15 deficient mice. So again, follow up from Dr. Sandoval's question, one of the clearest findings that we saw with semaglutide was that increased bile acid content, bile acids in both the genotypes. But again, high bile acid levels in the FGF15 knockout mice. Semaglutide also shifted the circulating bile acid composition, so not the total bile acids, but just the composition of species, against shifting it more to the secondary bile acids, such as deoxycholic acid. But then in the FGF15 knockout mice, the comparison between the chow versus the semaglutide treatment showed differences in several circulating bile acid species, such as the tauromuricolic acid, the hyalocolic acid, and the unconjugated muricolic acids. But then again, importantly, we saw a big change in the deoxycholic acid. So the granular profiling really revealed the semaglutide is not just changing how much bile acid was present, but it's really changing which bile acid species were present and also where they're located. Is it in the plasma, potentially the liver? We did not liver or the cecum, and how these patterns really depended on intestinal FGF15 or not. And this is really what we emphasize in our discussion, that it's the gut-liver lean mass communication. So the composition of the bile acid pool, especially maybe some of the changes that we see in these mouse models may be relevant to the lean mass outcomes than the total bile acid levels alone. Or maybe these changes are dependent specifically on FGF15.
I think this highlights something that we're understanding about the biology here, right? So every textbook says that bile acids are incredibly important for lipid absorption, right? They're vital to break down lipids so that you can push them around your body in an appropriate way. But it's super clear that they're signaling molecules, right? And that this work highlights some about how they can be signaling molecules, right? It's both the nature of how much bile acids you have and what types of bile acids you have. We know that these bile acids can drive endogenous GLP-1 secretion, as an example. They also are a way to reflect the microbiome. So the microbiome and what its constituents are, right, matter to which bile acids you're going to have, but also the levels of those bile acids will matter to which bugs survive in your GI tract as well, right? So it's a complex dynamic about how the biome, right, impacts the host physiology, and part of that has to do with its ability to push around the bile acids. And I think that ends up being a really important insight here, right? That that depending on how you lose the weight, you have different impacts on what it is that bile acid system is doing, and why it might be important for different aspects of what happens after you lose weight.
And while we're talking about how you lose the weight, this work follows on some prior studies that you had done that highlighted bile acids in VSG and in the role of FGF15 in
and also
that
the
weight loss.
body composition
And the
changes,
body
right?
composition. Thank you. Yeah.
Yes.
What Nadejda's previous work did, right, was to show that it's really important to regulate your bile acids, right? So you have a whole bunch of physiology that's built to do this, because after bariatric surgery, you get this big increase in bile acids. And this is work that Dar worked on as well, about, again, that change in the bile acids is a driver of that weight loss. But the question is, like, why doesn't it go too high, right? And the answer is it's because you get this increase in FGF15, which serves to reduce the amount of bile acids that are being produced. And when you get rid of that, the bile acids go up even more, and you have all these terrible outcomes. The way Nadejda explained it, she gave it a very clinical, clear version of this. But what really happens, right, is that the animals are a disaster. right? Their hearts go bad, their livers go bad, right? They lose more bone and more lean mass, right? So you get all these bad outcomes from vertical sleeve gastrectomy if it isn't accompanied by this protective response of FGF15. I think that sort of gets you into this idea about like, how do we use these systems, right, to produce weight loss that's sometimes called quality weight loss, right? So more fat and less lean mass loss. But also, how do you make sure that that's accompanied by positive outcomes, right? And that we limit the deleterious effects that can come with some weight loss under some conditions, particularly when it's really rapid, right? Like what happens after bariatric surgery.
So why don't we finish with any key questions, any other key questions you'd like to address moving forward? And maybe Nadia, we'll start with you because I think this is a little bit more of your baby, and then Randy can jump in.
I think future studies are really focusing on these observations in just this paper's broader conclusion of weight loss strategy and also the dietary context. How can we optimize, and just as Randy mentioned, quality weight loss, quality sustainable weight loss? How can we combine a lot of these treatment interventions to really optimize the body composition, again, the sustainability of weight loss, but also reducing the loss in lean mass or the skeletal remodeling? And I think a lot of these changes the answer to a lot of these different mechanisms lie in how the gut communicates to the liver in the outcomes. So this is really the focus of the follow-up studies.
From my point of view, one of the things that we do not have, right, are good biomarkers of who's going to respond well to different kinds of weight loss interventions. And when I'm seeing respond well, that isn't just about the amount of weight they lose, but also, again, about the quality and these other comorbid conditions. If we can get ourselves to the point, right, where we can use FGF15s and various bile acids, right, as markers for when things will go well and when things will go poorly, right, we're going to be way ahead of the game in trying to direct patients to get them to the right kind of intervention much faster than we do today. Obviously, in the case of pharmacology, we use the GLP-1 agonist as the assay, right? Do you lose weight and do you get better, right? We can't do that for surgery, right? Like that is a one-way trip. And so the question here, right, is can we figure out who's going to respond to which things are the best and make sure that we get people to the right interventions faster than we did before?
Well, with that, Randy and Nadia, we want to thank you for joining us today to share your expertise and for discussing your recent work on the roles of FGF15 in lean mass preservation during weight loss. We encourage the listeners to read the full article in the June issue of Diabetes.
Thank you so much for having us.
Thank you for having us.
Welcome to another episode of Sweet Talk. I'm Kevin Williams, and I'm joined by my co-hosts, Dr. Darleen Sandoval and Dr. Kirk Habegger. Today, we have a special guest who has spent his career asking fundamental questions in metabolic biology, like how does the brain know how much fat the body is carrying and what does it do about it? That question led him from the molecular dissection of leptin receptor signaling all the way to defining specific hypothalamic and brainstem circuits that are regulating feeding, glucose homeostasis, and body weight. Our guest is Dr. Martin Myers, who is the Marilyn H. Vincent Professor of Diabetes Research at the University of Michigan, director of the Elizabeth Caswell Diabetes Institute, and a former editor-in-chief of this very journal, Diabetes. He is also the recipient of the American Diabetes Association's 2010 Outstanding Scientific Achievement Award, which recognizes his seminal contributions to understanding how leptin signals are translated into physiological action. Martin, congratulations on that recognition, which is, I guess, now 16 years ago when you received that award, and welcome to Sweet Talk. It's great to have you.
Thanks, Kevin and Dar and Kirk. It's great to be here.
So we'd like to start these off with a little bit of an icebreaker question, and I think I'm going to ask you about your hobbies and interests that might be outside the lab. I think I might know an answer to one of these, but is there something specific that you like to do outside the lab that might help you reset and recharge?
And I assume you're not talking about reviewing papers in front of the TV at night. No,
To
so
each their own, you know.
as you know, I rowed in college and then took a bunch of years off. But for about the past 20 years, I've been rowing with the Ann Arbor Rowing Club here in Michigan, and you know that keeps me good and tired, so I fall asleep in seminars at appropriate times.
And still wake up to ask the most potent question.
Yeah, I wake up when the clapping starts. But yeah, I'm on the water 5 or 6 days a week. I mean, not in the winter when we're on rowing machines, but it's a good way of flushing your brain, right? Because you really can't focus on anything else when you're rowing.
You all travel as well, right?
We
What
do.
do they call, what do they call, regalas? Is that what they
Regattas.
call them?
Yeah,
Yeah.
exactly. So most of them are relatively local. You know, we're in Michigan, and so we go to Michigan regattas, Indiana, Ohio, etc. And then once a year we make usually a longer jaunt and go off and do some rowing at the head of the Charles.
And when you say we, have you recruited people within your lab? Is it a requirement when they join that they have to try out? What's going on there?
Definitely bonus points there. I've had a couple people in the lab start rowing, and for some people it's stuck, and for some it hasn't. But that's relatively the minority. I do have a couple of diabetes focused faculty colleagues who row with me, though. So Dave Olson, who's a pediatric endocrinologist and also one of my close collaborators, and then also Paula Goforth, who's an electrophysiologist focused on a lot of the same circuits that we are, also rows. So I see them bright and early or dark and early most mornings.
Nice. And I love it when that word electrophysiology makes it into the podcast from time to time. More of that, please.
Yeah. Well, ask me the right questions.
So Martin, I get the first more, I guess, science/career targeted question. And we're going to go back to your training with Maury White that was focused more on insulin signaling. So how did you make that journey from insulin signaling to leptin and hypothalamic circuits?
I wish I could tell you that there was a grand scheme involved.
Yeah.
But so I was an MD PhD, and when I got done with the MD part, I realized that I was a researcher at heart and not a clinician at heart. So I went back to the Joslin Diabetes Center, and they were nice enough to give me the opportunity to sort of start my own research program. The rule was, though, that I had to work on something other than insulin signaling because it's what I had worked on as a graduate student. So I spent a few months casting around, trying to figure out how I was going to leverage my knowledge of tyrosine kinase-based signaling and do something metabolically related that wasn't insulin. And it turns out that leptin and the leptin receptor had just been cloned, and I was like, "This is it! This is a
Ding,
great
ding, ding,
opportunity."
ding.
Yeah.
That's great.
You know, so as you probably know, we worked on leptin receptor signaling as a sort of a problem in cell signaling for a couple of years. And then we started dipping our toes into the mouse world and physiology by mutating some of the signal transduction pathways for the leptin receptor. And at that point in time, we realized that if we're really going to understand what leptin does, we have to understand more than just what it does in the cell. We have to actually understand the neural circuits on which it works. And that's been, I guess, the past 25 years or so of my life.
Yeah. So it's interesting, like in the late 90s, the sort of boom about leptin, at least from a and maybe even like at least for the hope for it didn't pan out, but there was certainly hope for a treatment angle to leptin at that time. It felt like, and I've heard other people talk about it being a very exciting time, and it feels a little bit, is there a corollary between what we're seeing now with obesity treatment in terms of the excitement about obesity research and sort of a field making a little bit of a change?
It's true. Back in the late 90s and early aughts, it was just amazing. You know, you'd go to a Keystone meeting or to the ADA meeting and sit in these symposia, and you would just be on the edge of your seat all the time because things were happening so quickly, right? It was just extraordinarily exciting. And I mean, for me, it's been exciting all the way along, but you're right that there are times of really rapid progress And that was one. And also we're certainly in another with people trying to figure out how all of these incretin-based therapies and some of these other emerging anti-obesity medicines are working. There's just a ton happening. I'm back on the edge of my seat at Keystone meetings.
So Martin, I want to maybe follow up on something you mentioned when you were answering Darleen and talking about dissecting leptin signaling. And some of your earlier, very elegant work utilize specific point mutations for specific phosphorylation sites. Can you walk us through that logic and why that approach was necessary and what was surprising about it as you saw that data being uncovered?
Okay, so coming into leptin signaling, the advantage I had was sort of really understanding the biology of tyrosine kinase signaling and the way that works. One, let me take a step back. So most people think about kinases and they think about serine threonine kinases and how phosphorylation changes the activity of an enzyme, right? Like a kinase. But it's different for tyrosine kinase signaling. There are some activating phosphorylation events, but mostly what happens is the kinase phosphorylates tyrosine residues either on the tail of the receptor itself or on a substrate. And that doesn't change activity so much as it creates a binding site for downstream signaling proteins that have these specialized phosphotyrosine binding domains. Many of them are SH2 domains. And so that creates an opportunity for really dissecting downstream signaling in a way that you wouldn't necessarily be able to do if you were working with the serine threonine kinase, because you can mutate a single tyrosine residue, and that prevents the recruitment of a specific singular downstream signaling pathway. And using that, we were able to define first in cell culture and then in animals the importance of STAT3 signaling to everything, well, most everything that leptin does, and then to define roles for some of the other tyrosine phosphorylation sites, including one that's involved in feedback inhibition.
Yeah, and when you look at the signaling map that you helped build, what do you think still remains most underappreciated about how the leptin receptor actually works?
Oh, I mean, so there are a bunch of things. The one that still most interests me and sort of has me pounding my head against the wall, I think relatively frequently, is well, it's clear that STAT3 signaling is really important for leptin receptor signaling and the control of physiology. There's clearly an important second signal out there. And although we've sort of been banging on it for 20 years, we're not sure what it is. And as far as I know, nobody else is sure either. So that's a, that's a real problem. I guess the other thing that I'll say is one of the major limitations that we still have in terms of understanding leptin receptor signaling is that we've got great cell culture systems for looking at leptin receptor signaling. But it's very, very hard to detect leptin receptor itself at endogenous levels, for instance, in neurons. And with the exception of STAT3 phosphorylation, it's very difficult to look at direct leptin signaling, in cells in the brain. And so, we don't really have a good sense of how the receptor is trafficked, whether it's, degraded, internalized and degraded after, it gets turned on by leptin or what. The cell biology of the receptor itself, in a context that really matters, it remains pretty much a black box.
And Martin, I want to pick up from there. I have to say, there's no way you would know this, but that Sarah Bates paper from 2003, where you showed the segregation of STAT3 signaling as required for energy balance and not reproduction, that was one of the first papers I had to dissect in graduate school for a journal club. And I still reflect on it. It really struck me then of how clean the logic was of just that one amino acid substitution, one disrupted signal, and suddenly you could read out which physiological functions were STAT3 dependent and which weren't. It was really kind of a masterclass of genetic dissection of leptin receptor signaling. And I think you carry that on with much of your work even today. And so I definitely follow your work for that kind of masterclass definition of what's going on molecularly, but then also now moving on into circuit dissection, as it relates to leptin, going from lateral hypothalamic leptin receptor neurons projecting elsewhere, sexually dimorphic PMV neurons and others. What was the intellectual shift for you to move from that molecular physiology to circuit biology? How did the tools available to you change the questions you were asking? Because, the technology definitely goes hand in hand with the type of questions that we can ask. Over the years, what have you found from those technological innovations to drive the science?
So I guess the thing that really drove the switch for me and the lab was when we generated a leptin receptor Cree mouse. We generated this with the idea that we were going to be able to delete specific signaling molecules from leptin receptor neurons and do more of our cell signaling stuff. And we did do some of that. But when we crossed that leptin receptor Cree onto a reporter background and peered into the brain and saw all of these leptin receptor cells all over the place, it sort of made us take a step back from the AGRP and POMC neuron centric view of leptin action that was prevalent at the time, including sort of in our own hearts and heads, and made us realize that if we were really going to understand leptin action, we needed to understand it as more than just how leptin turns on this particular signal or that particular signal, but what all of the different types of leptin receptor neurons do, and then presumably how leptin acts on each different cell type, because presumably, leptin turns on, for instance, a pattern of gene expression in POMC neurons that's different than the pattern of gene expression that you would see in, say, the leptin receptor GLP-1 receptor neurons that you and I have both been playing around with recently. And so that's sort of what drove the process. And obviously, we're still running down that rabbit hole, right? There are still more types of leptin receptor neurons that we've found that we don't know what they do, and really understanding how leptin signaling modulates different types of leptin receptor neurons, I think is the next step that we have to take.
And there's been a classic debate in the field with leptin as to, is it a signal of energy surplus or is it a signal of energy deficit? And I'm curious to get your thoughts of what you might tell the audience, the primary drivers of leptin signaling and physiological context that it might be playing.
Okay, so I think everybody would agree that if you go from zero leptin, like in a starved animal or in in a leptin deficient animal, to normal physiologic levels of leptin, there are a characteristic set of changes that bring physiology from a starvation response to normal, right? And then the question is, how is it different when you go above normal? And certainly it's different in that, if you go above normal in levels of leptin, you don't see that excess leptin driving big differences in food intake and body weight the way you would if you go from zero to some leptin. One of the things that has been really empowering for my lab over the past 5 years or so is a collaboration with a guy named Tuna Pers, who's at the University of Copenhagen in Denmark. And working with him and so he's a bioinformatician extraordinaire who's really interested in a lot of the same circuits and questions that we are, but he attacks it from a different perspective. And one of the things that we've learned with and from Tuna is that really leptin action on different leptin receptor cell types can be differentiated. And this paper isn't out yet, but it turns out that different leptin receptor cell types respond differently to elevated leptin. I mean, most of the cell types, again, when you go from zero to normal leptin, big transcriptional response, right? But when you go from normal to above normal, like in obese animals, first of all, the response of different cell types, the transcriptional response tends to be more varied. And some cell types respond extraordinarily robustly to the excess leptin, and some just don't respond at all, right? There's just, so for AGRP neurons, for instance, you add extra leptin and you don't really see much happening. Whereas in the leptin receptor, GLP-1 receptor neurons, they respond like crazy. And one of the questions I think is, how does that happen? And B, what does it mean that, for instance, an AGRP neuron doesn't budge in its leptin responsiveness when you go from normal to obese?
Yeah, I love that cellular description of the leptin kind of resistance that people talk about, but this is at the cellular level. And I know that you've done work on trying to define actual leptin resistance versus what might be happening to these pharmacological insults, or possibly in what you're talking about, these different levels of energy states and different nuclei that may have this resistance, and in quotes, and others that might be more sensitive to leptin throughout the dynamic range of lower levels to higher levels. And so it's really interesting work that I think a lot of people in the audience are probably following along and wanting to find out more. So I think it's great insight, and we'll be looking forward to that paper that will be coming out soon, I hope.
Me too.
So, Martin, you've talked a bit about the heterogeneity of those leptin receptors, but one of the things that I think would be really interesting for maybe people that are thinking about going into the brain, maybe graduate students or postdocs, is to talk a little bit about the strategies that you use to try to understand the heterogeneity. And then I know the listeners can't see this diagram, but I'm going to show it for Kirk and
Wow.
for Kirk and Kevin. This is a famous Martin diagram of the heterogeneity of the AP and NTS, and including the yummy and yucky circuits here. I wish I would have gotten it signed before.
Yeah, you should have. I was going to ask you next. Did you get an autograph?
Yeah, but I think one of the things that's really cool is that you really do incorporate a lot of different strategies to try to answer these questions. So maybe talk a little bit about that.
Sure. I guess maybe I'll start by saying, so when I was back, doing my graduate work, I had I had a bunch of housemates, but one of them was another MD PhD student who was a neuroscientist, and I used to ridicule him unmercifully because I didn't understand why anybody would work in the brain, given how primitive the tools were. Right? It's like, I mean, you can't learn anything by like buzzing this area or buzzing that area, because everything is every nucleus is so heterogeneous, especially when you get into places like the hypothalamus and the brainstem. And the thing that I think has really made the difference, not just for me, but for the entire field, is the molecular genetic tools that we have to identify and manipulate very specific cell types so that we can understand not only what happens if you turn on all of the cells in the nucleus of the solitary tractor and the AP, but you can define different cell types and their receptor component and their downstream targets and so on and so forth, and really start to tear apart the circuit basis for all of these behaviors that we care so much about. Yeah. So I can't ridicule neuroscientists anymore.
And Martin, I want to talk about, you alluded to this a little bit earlier when we were talking about that leptin energies surplus deficit conversation of GLP-1 receptor expressing neurons that co-express leptin receptors. And then you also have many ongoing projects right now as it relates to hindbrain GLP-1 receptor signaling. And I was curious if, I know that there is a paper that you have out that's a bioarchive, a preprint right now. I don't think it's been accepted or published in any fashion outside of that preprint, but it does describe some aspects of GLP-1 pharmacology in the sense that these compounds can induce satiety. They can also induce aversive effects, nausea, sickness, and potential overlap of circuits that might be targeted by these pharmacological compounds, and whether or not you can dissociate those aversive versus satiety type signals. And I was curious if you could maybe inform the audience a little bit about what you're finding in that area, maybe something related to that preprint, and kind of walk us through what's going on.
Sure. So I guess specifically focusing on GLP-1 receptor neurons and obviously GLP-1 receptor agonists, which are sort of in the process of revolutionizing the treatment of obesity and type 2 diabetes. There are a bunch of different types of GLP-1 receptor neurons in the brain, right? And most of them lie behind the blood-brain barrier. So for instance, this population of neurons that you're alluding to in the nucleus of the solitary tract, the ones that you and I both care about that live in the dorsomedial hypothalamus, and there's some in the lateral septum that people talk about a lot. And then there's this population of GLP-1 receptor neurons that lies outside the blood-brain barrier in the area postrema. And that one is the one that not only can drive down food intake, but also turns on these sickness responses like nausea and so on and so forth. And most of the neuron populations that have GLP-1 receptor neuron populations that have been looked at that live behind the blood-brain barrier, including those in the nucleus of the solitary tract, really drive suppression of food intake that looks more like satiation than the ones in the area postrema. But it seems like what we're finding anyway is that the GLP-1 receptor agonists that are out there today do a much better job of accessing and activating the area postrema GLP-1 receptor neurons than the ones behind the blood-brain barrier. And unfortunately, that means it is probably pretty hard to dissociate the nausea from the suppression of food intake. Although it does make one wonder what would happen if you were able to drive GLP-1 receptor agonists into the brain, do a better job of accessing all of those other GLP-1 receptor neurons. Would that give you a drug that's less aversive because you're able to activate those cells that are inside the blood-brain barrier, as well as the ones that are in the area postrema, maybe letting you dial back doses or something like that.
Yeah, and there's been concepts of biased agonism at the receptor level that might help to separate the efficacy of from these side effects, or maybe other types of brainstem targeted approaches. You have the combination therapies of GLP-1 GIP, which seems to have a little bit less nausea and induction due to the compounds. Do you think those types of strategies are where the field is going to stay at? Is there another strategy that might be more useful in trying to target those satiety versus the aversive actions without, I guess, giving away too much of your secrets?
There are no secrets, as you know. Any piece of data I have, I'm going to bend somebody's ear about. But I guess for the past few years, we've been very interested in a set of neurons that live inside the area postrema. These, it's a single population in terms of tuna pairs bioinformatic definition. Some of the neurons in there express the GLP-1 receptor, some express the calcitonin receptor. There's some other receptors in there. And the nice thing about those is that they seem to turn on satiation-like responses as opposed to sickness-like responses. And I think strategies that target those neurons would be much, much better tolerated, at least from a nausea standpoint. And we think that that's probably at least part of how these amylin receptor agonists, or the dual amylin receptor calcitonin receptor agonists, work. So hopefully there are additional ways of targeting those neurons as well. But if I wanted to lose weight and not feel nauseated, that's probably where I'd start.
Martin, since this is a podcast for diabetes, I thought maybe we talk a little bit about glucose. And specifically, your work comparing STAT3 and JAK2 animals, and how there are direct effects of leptin and its regulation of glucose homeostasis. Can you kind of take us through the details of that work?
Sort of taking it back to the beginning, I think there's a whole bunch of really nice data, starting in preclinical models from even before perhaps leptin was cloned, suggesting that the absence of leptin, or the absence of leptin receptor signaling, causes the dysregulation of glucose homeostasis. And obviously the question is, to what extent is that physiologically relevant, and to what extent, and how does that actually happen? And we could spend an hour talking about each of those, but my take on it is that it's physiologically relevant, probably where leptin matters the most, which is again between zero leptin and normal leptin. And so in people with uncontrolled type 1 diabetes, that's a super important space, right? Because leptin turns out to be very low. In terms of, is there a role for excess leptin, when you go from normal leptin to obesity in the control of blood glucose? I think the data are maybe a little bit murkier. I mean, Philip Scher has some interesting data to say that excess leptin may actually be bad in obesity. And I think the only way we're going to figure this out is by actually understanding how leptin controls blood glucose and all of the components of blood glucose. And of course, the problem is now we're in the brain and we've got a zillion types of leptin receptor neurons, and each of them has a contribution. So
Right.
Joel Elmquist and Kevin have done a bunch of stuff with POMC neurons and HRP neurons and shown, and a whole bunch of other people have worked on this as well, shown that those are actually an important part of how leptin regulates glucose homeostasis. But there are clearly other things going on too. We defined some neurons in the midbrain, which are really important for the counter-regulatory response. And I'm sure there are others. And it's just going to take a lot more work to define what leptin's doing on each of those circuits and why, and how that contributes to physiology and pathophysiology.
So, Martin, since this is a podcast for the Diabetes Journal,
Mhm.
we'd love to hear about your time as editor-chief of Diabetes, and I'm just curious whether that role made you think at all about sort of our current, maybe where the field is headed, or even the state of the peer review process and our publication processes. Any thoughts on that?
Oh, I mean, lots of thoughts on that. So,
Yeah.
first of all, I mean, it was an amazing opportunity. I was thrilled to do it, even though it turned out to be, an absolute ton of work. But I think, the breadth of papers, just like the breadth of diabetes research, was really enlightening. I mean, I felt like I learned so much about things that I wouldn't really have paid as much attention to otherwise, just because, when you've had, you know, 6 or 10 or whatever it is, papers coming across your desk every day, it's a lot that you get exposed to.
Yeah.
In terms of what I learned about the direction of the field, remember that I stopped being editor in chief about 5 years ago. So back then, I could tell you that incretin agonists were going to be really big.
Good call.
In terms of where things are going forward, asking the current editor in chief, Dave D'Alessio, would probably be a better idea.
Yeah.
In terms of the journal, the publishing world, we're obviously in a very different space than we were 20 years ago, right? 20 years ago, all of the big journals, most of the big journals were published by scientific societies, like as diabetes is, and endocrinology, and so on and so forth. And the proliferation of commercial journals or for-profit journals or whatever has, for a while, was really eating the lunch of the society-based journals. I think we've established a new equilibrium, and each of the journals, again, has its own space. And I think it's great to note that diabetes, I think, has done very well in this process. It remains, one of the linchpin journals in the diabetes and metabolism space.
Martin, I think we all thank you for your service to the society, to the journal. Also, we thank you for joining us today to walk us through the molecular dissection of leptin receptor signaling all the way up through brainstem circuits that help explain how these GLP-1 based, incretin based drugs are actually working and helping to find weight loss versus nausea separation and those effects for the field. So before we let you go, where can the listeners follow your work? Do you have a lab page, an institutional page? Are you on social media? Anything you could point them to?
So, I mean, the short answer is no. I actually do try to stay away from social media on purpose. I think if you want to follow my work, the best thing is to, follow publications, follow bioRxiv, or if you have a question, just shoot me an email. I'm always happy to talk about science.
Nice. So thanks so much for joining us. And for our listeners, as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks, you guys. This was fun.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.